Cancer is one of the leading causes of mortality worldwide, with over 14 million new cases diagnosed and over 8 million cancer-related deaths each year. But while the incidence of cancer cases increases, there is reason for hope. Advances in technologies, such as next-generation sequencing and gene editing, have led to the development of treatments that are extending patient lives. In particular, the development of effective immunotherapies for a range of cancers has been an area of great interest in recent years.
Immunotherapy as a whole seeks to empower the patient's own immune system to not only find the cancer, but also destroy the cancer cells. And these therapies come in different formats. These could be in the form of engineered immune cells, or it could also be in the form of checkpoint inhibitors. These are primarily antibodies that seeks to unleash the power of the immune system to recognize and destroy cancer cells.
Immunotherapies work by stimulating the body's own immune system to recognize and target cancer cells. However, cancer cells can learn to evade these immune cells. They can evade attack by using immune cell brakes, called immune checkpoints, which prevents the immune system from recognizing the cancer cells. Checkpoint inhibitor therapies work by releasing the brakes, targeting molecules that are involved in activating or inactivating the immune response, and thus allowing the immune system to attack cancerous cells. The 2018 Nobel Prize in Medicine was awarded to two scientists who pioneered this new way to treat cancer. In the 1990s, when Dr. James Allison was at the University of California, Berkeley, his team discovered the checkpoint protein CTLA-4 and engineered an antibody that could bind to CTLA-4, removing the brakes on T-cell activity and unleashing them to attack cancer cells. Working independently, Dr. Tasuku Honjo at Kyoto University in Japan discovered PD-1, a protein expressed on the surface of T-cells that plays a role in downregulating their activity when it meets a cell that expresses its ligand, or PD-L1. While this is important in preventing autoimmune diseases, if a cancer cell expresses PD-L1, it can escape being targeted by T cells and will grow unchecked. With the addition of anti-PD-1 and PD-L1 antibodies, the immune checkpoint brakes are removed and T cells are able to attack the tumor cell. In recent years, several checkpoint inhibitor therapies have been approved by the FDA. Clinical trials for checkpoint therapies against most types of cancer are now being conducted, and scientists are testing numerous other checkpoint proteins to see if they could act as targets. Dr. Robert Prins, a professor at the David Geffen School of Medicine at UCLA, is focused on clinical medicine and translational immunotherapy for malignant brain tumors.
The advent of all different types of immunotherapies that are out there, we're doing quite a bit of the assessment of T-cell function inside brain tumors, trying to understand what influences the effectiveness of vaccines, or how do T cells become dysfunctional in the brain tumor microenvironment. And that spans the boundary of many different types of studies, and that includes checkpoint blockade and other agonistic antibodies that enhance T cell function together with vaccines. If you add a vaccine with a PD-1 blockade, you can enhance the infiltration of tumor-specific T cells into the tumor, and if you purify those, you can see enhanced killing of the cells.
While immunotherapy discoveries are revolutionizing cancer treatment, there are still many challenges with developing these therapies, including trouble predicting efficacy, unpredictable side effects, and the high cost of these types of treatments. Researchers are working diligently with the help of new and evolving technologies to address these issues, but there is still much work to be done.